All right, that's our official warning. The folks at Apple telling us it's officially 9 o'clock on the East Coast here. Thank you everyone for joining us for the first webinar in a four-part series that's going to explore topics in understanding and managing periosystemic health and oral biofilms. Sponsored by our friends at Sunstar, this series is offered expressly for you as dental students and members of the next EDS. I'm Rich Groves. I'm the Vice President of Academic Affairs for the organization, and it's my pleasure to introduce you to our speaker for this evening, Dr. Greg Kurtzman. He and I have known each other for a number of years, and he's really a terrific educator, author, authority in this topic. He's published over 500 articles in the dental literature, international journals here in the States, on topics ranging from periodontal health, bone grafting, dental implants, former faculty member at University of Maryland, and really an authority who has done a tremendous amount of research and investigating topics just like the one we're bringing to you this evening. Just before we get started, I just want to cover a few housekeeping points. My name again is Rich, and if you have any questions, I'll be your moderator for this evening. And if you have questions you'd like to queue up for Dr. Kurtzman, we're going to address as many of them as we can after about 45 minutes of his presentation. You can use either the chat feature in your window or the Q&A section, and we'll queue all of those for just after the didactic portion, and we'll get Dr. Kurtzman to answer as many of those as we have time for this evening. So we encourage you to ask as you go. We'll get to as many as we can, and without any further ado, it's my pleasure to introduce you to our speaker this evening, Dr. Greg Kurtzman.
Hi, and welcome to the presentation. We're going to be basically talking about oral biofilm and systemic connection, and that's me, just a little bit about me. What I think is kind of interesting about this topic is that I started dental school in '82, and prior to that, I think we were basically tooth mechanics. We were taught how to repair teeth, and anything past the back of the throat we didn't concern ourselves with. When I got into dental school, they started giving us a little bit more medical connection. So that we basically had some information that what's going on in the mouth really had an effect, or vice versa with the systemic issues. And I think that probably in the last 15 to 20 years, I think that's gotten better for the dental students because you're getting more training as healthcare practitioners versus just being a tooth mechanic. So basically, this is going to be a four-part series, and tonight we're going to basically talk about oral biofilm, inflammation, and the oral health relationship. We'll do part two, which is systemic effects, and part three is some more systemic effects in different parts of the body. And part four, we're going to finally pull it all together and say, how do we recognize this stuff, how do we treat it, and how do we prevent dental and systemic issues? And I think that this is really important, and if you look at the literature, and we'll show some of that in the presentation tonight, there's a lot of connections that are coming out, and it's an accelerating amount, connecting what's going on in the mouth to what is going on in the rest of the body. So basically our agenda is we're going to talk about etiology, we're going to roll that into pathways, into the role of biofilms and bacteria, how the systemic effect is, the host response, and finally in the last of the series we'll talk about management. So let's start tonight's presentation. Oral biohealth, inflammation, and the oral health relationship. So basically, periodontal disease are a group of diseases affecting one or more of the periodontal tissues. And most of the people in the United States, at least, 47% of adults have some form of periodontitis. And that could be just bleeding occasionally when you brush to having bone loss and mobile teeth. So 70% of U.S. adults age 65 and older have periodontitis. So as we get older, we're seeing more of this, and of course, as we get older, we're seeing more systemic issues, more cardiac, pulmonary, diabetes, high blood pressure, all kinds of other systemic issues. And we found that the periodontal problems and caries are connected with plaque and oral bacteria, and increasingly with the systemic health. So it's kind of interesting. We basically have to look at it this way. Oral bacteria, what we used to call plaque when I was in dental school, we've reclassified, and we've sort of followed in medicine where they're basically talking about accumulations of bacteria as really a biofilm. And basically what we've done is we've focused on their effects within the oral cavity. Accumulator research has established a direct link between oral health and systemic disease, over 200 possible connections according to the ADA. Harmful microorganisms are always present in periodontal disease, and specific patients are predisposed to harboring these oral organisms and increased systemic health risks. And as you get out into practice, and you may be seeing that in dental school, you're going to get patients who come in who do a meticulous job brushing. You never find any plaque, calculus, or anything, and yet they're still having periodontal issues. And then you're going to get patients, as we used to say, they don't know what the fuzzy end of the toothbrush is. Their teeth are completely covered in plaque and calculus, and they just never brush, and they have no health issues whatsoever. So a lot of this really falls into how our systems are susceptible, and some people are more susceptible to things than others. So the accumulator research is evidence links periodontal disease and chronic oral inflammation in a multitude of health conditions, including respiratory disease, diabetes, osteoporosis, and many more things that we'll cover as the series goes on. Plaque today is recognized as a complex environment, and we've returned that oral biofilm. So as we wait for Rich to do this poll, basically we really have to look at there's a lot of things that go on in the mouth, and there's the poll, so I'll stop for a moment.
All right, everyone, thanks very much. When you consider, when you're managing your future patients, let's just take a quick pulse to see what do you imagine to be the most daunting aspect of your caring for your future patients. All right, those answers are already rolling, thank you. Don't be bashful.
There's no wrong answers.
There we go. Interesting. Okay, patient communication and education. I'm going to promote this poll right on screen so that you can all see the same. There we go. Any surprises here for you, Dr. Kurtzman?
No, I would agree with this. I think patient communication is important. Consistent treatment is also. I think that's, if I were answering this, I would have picked A myself.
Excellent, excellent. Well, thank you very much. Please continue.
Get rid of that, the box.
Coming right down.
So what is oral biofilm? Plaque is a community of microorganisms found on teeth surfaces and within the sulcus, which are embedded in a matrix of polymers of host and bacterial origin. There's over 700 different species of bacteria naturally reside in the mouth. And these are going to vary. You can have bacteria in one tooth, and the pocket next to it and the next tooth has totally different bacteria. So you're not going to necessarily find the same bacterial species throughout the entire mouth causing problems. And it's not going to usually be due to just one bacteria. It's going to be a multitude of those. And the mouth is a perfect inoculator for bacteria, which can double in number in just hours if left undisturbed. Bacterial proliferation creates an intricate network of protective layers, which is a matrix. It's pretty much a slime matrix that protects it, and channels that develop into a biofilm are a major cause of periodontal disease. So how do we manage these pathogenic bacteria? Bacteria and biofilms are less susceptible to antimicrobial agents, either locally applied or systemically administered. And basically, when I got out of dental school in '86, the direction was basically placing into the pockets antibiotics. And you basically either treat the entire arch or the quadrant that you were treating with something, or you just do isolated pockets within that in the hope that those antibiotics were going to basically kill the bacteria and allow the host to basically regenerate the tissue. And these microbial communities can display increased pathogenicity due to this synergism that goes on between the bacteria. So the structure of the biofilm may restrict penetration of antimicrobial agents, that slime really basically blocks it out, while bacteria growing on the surface are susceptible to antimicrobial agents. So if we have stuff growing on the outside of the tooth, on the enamel, that's very susceptible to antibiotics because there's really no slime matrix involved with that. But if you basically have it in the sulcus, they're basically encased in the slime matrix, and it's sort of a protective mechanism, and it basically helps really make those that colony of bacteria immune to anything that we're placing into the pockets. Aggregated bacteria work together as a community. producing specific proteins and enzymes by way of quorum sensing, and utilizing oral fluids as the vector for transmission. Bacteria in these oral environments have evolved as part of a multi-species biofilm and may require interaction with other bacterial species to grow, forming complex bioenvironments. So this is much more complex than we were taught, at least when I was in dental school, and we understand it a lot better, and really a lot of this research is coming out of the medical end. Bacteria in the biofilm have the ability to regulate numerous processes through quorum sensing. Quorum sensing is a cell-to-cell communication mechanism that synchronizes gene expression in the biofilm in reaction to the density of the cell population. So as the cell population increases, the bacteria basically start working together by communication, and they basically turn things on, turn things off in order to make the community thrive and grow. This includes secreting specific enzymes to activate or deactivate the genes of other bacteria. These bacterial byproducts provoke an immune response which recruits white blood cells to the site to kill the invading bacteria. This results in localized inflammation of the surrounding gingiva. Via quorum sensing, the bacteria have the ability to confuse the defending WBCs chemotactically by releasing chemicals into the local environment, rendering the immune response ineffective. That's why basically we'll see in patients that have advanced perio or long-term perio that the body is just not fighting it. And sometimes, as we'll go into, the body is being fooled into thinking that other parts of the body, the bone and gingival tissue, is actually foreign substances and is actually attacking itself, and that increases the bone loss in these cases. As we will note, WBC's white blood cells have a 3-day life cycle. If they don't engulf bacterium and destroy it within that time frame, they lyse and die. So the components within those white blood cells that are intended to kill the bacteria are now available to damage the tissue they were meant to protect, contributing to bone loss, deepening periodontal pockets, and inflammation. And a lot of this has to do with how reactive the patient is. It's just a perfect example, poison ivy. Well, it's not a dental thing, but we can take a group of people and we can rub poison ivy leaves on their arm, and some of them will react a lot, and some won't react at all, and people will be between those two spectrums. It's the same thing with how the body reacts to these bacteria and the bacterial products. Sometimes we're going to see a lot of bone loss, and sometimes we're not going to see any. So quorum sensing has been, let me see if I can remove this one thing here. Quorum sensing has been described in both gram-negative and gram-positive bacteria, the basic mechanism that controls gene expression in both groups of bacteria. And I try to basically give you some science to prove or show that this is really not an opinion, it's basically well-researched at a peer-reviewed journals. Let's see if I can get that back going. There we go. When quorum sensing bacteria are growing, they produce and release to the external environment a series of molecules called autoinducers at a low basal level. As the population increases, these molecules accumulate until they reach a certain threshold level, which leads to the activation of different sets of target genes that allow the bacteria to survive environmental changes. So what happens is bacteria that would normally die under certain circumstances, when you put them together into a biofilm, they basically help each other by, as we said, turning genes on and off, and they basically become they really survive a lot of environmental changes. Since bacteria within the biofilms reach a high density, it has been suggested that quorum sensing might play a key role in bacterium-to-bacterium communication, and therefore in the formation of biofilms. Quorum sensing helps bacteria to communicate with each other and in coordinating their behavior. Many diseases of human, plant, and animals are mediated by communication through quorum sensing. Quorum sensing is a process of chemical communication among bacteria, and is defined as gene regulation in response to cell density, which influences various functions such as virulence, acid tolerance, and biofilm formation. Quorum sensing relies upon the interaction of a small diffusible singular molecule with a center of or transcriptor activator to initiate gene expression for coordinated activities. The biofilm can be disrupted by alarming the quorum sensing mechanism, utilized by various species of bacteria that together form the black biofilm. The inhibition of quorum sensing is commonly referred to as quorum quenching. So basically, we have quorum sensing inhibitors, which is lactinase, actylase, and some of these other things, human hormones and other compounds that will basically inhibit quorum sensing, that communication between bacteria in the biofilm. We also have quorum sensing inhibitors such as horseradish, garlic, turmeric, and cumin. Turmeric and cumin is interesting. There's been a lot of research that's coming out that sort of plays on decades and decades of herbal medicine, that it is an anti-inflammatory, and they're finding out that it does help inhibit these products of quorum sensing. Citrus flavonoids, grapefruit extract, nutmeg, and sweet basil are just some of those. So quorum sensing biofilms are distinct properties. At high cell density may provide protection to the biofilm bacteria and influence community structure by encouraging the growth of beneficial bacteria to the biofilm and discouraging the growth of competitors. A biofilm environment confers certain properties to bacteria that are not seen in the normatic state. Recognizing dental plaque as a biofilm and not as a bacteria in the planktonic state. Or planktonic, when they say planktonic state, what they mean is that bacteria that's stuck to the enamel of the tooth. It's in a flat surface, it's not protected, it's open to the environment, where a biofilm we have to look at as being protected, it's in the sulcus, it's in an area that basically is very hard for the body or us to get to affect it. Biofilms are microcolonies of bacterial cells, 15 to 20% by volume, that are not randomly distributed in a matrix. And the matrix is about 75 to 80% by volume. Bacteria and biofilms cluster together to form colonies, and each microcolony is an independent community, and site to site, this will vary even adjacent sites. As we said earlier, you can basically test the bacteria and culture it in, let's say, the buccal sulcus of a lower first molar, and the second premolar next to it, you can do the same thing, and you'll find that totally different bacteria site to site, tooth to tooth. And sometimes, in some cases, you may have different colonies on the buccal and the lingual of that same tooth. So that basically, just because they're in an area doesn't mean that everything is uniform throughout the area. So as we see here, we look at biofilm from adjacent pockets. These are two basically samples that were taken in adjacent pockets, and we could see, looking at this, the bacteria on the left looks totally different from the bacteria on the right in those images. So we basically have to look at something. This is just not we're attacking one type of bacteria. We basically have to deal with 700 or more different species that are potentially in the mouth. Function of the biofilm. Outer layer is high concentrations of oxygen and nutrients, resembles planktonic bacteria. Those are aerobes. Slough off and attach at other sites, forming new biofilms elsewhere in the mouth or body. We have a mid-layer where the organisms can downregulate their metabolic activity, which means that they basically go in almost into a hibernation state and can utilize nutrients and exchange genes that have potential for drug resistance. So we might be able to put a drug in there that's going to affect the outer layer, but once it gets to the mid-layer, it really has no effect. And then we have the inner layer, which is attached to the substratum and is the earliest part of the biofilm, the oldest. Effectively downregulated, at least metabolically active, and most of it is fossilized. So most of the bacteria in this area, because of the mid-layer and outer layers, is just basically hibernating in a sense. So this provides inheritance for future populations that transfer laterally. So if we look at plaque as a microbial community, Marsh and Bradshaw in 1999, so that's a long time ago. Bacterial interaction in the community can be beneficial or antagonistic to the interacting species. Beneficial would be enzyme compilation, food chains, co-aggregation, and activation of inhibitors, such as antibiotics, subversion of host defenses, and antagonistic could basically be producing bacteriocins or hydrogen peroxides that will basically destroy neighboring bacteria. So if we look at the composition of the biofilm, the bacterial community in the biofilm is diverse, with variations in many species being detected, which can be different from site to site in the same patient. And I realize that we're repeating this stuff over, but it really needs to be repeated in order to understand it. because once formed, the species composition at a site is characterized by a degree of stability among the component species due to a balance between synergism and antagonism. Slime matrix, the backbone of the biofilm, is basically exopolysaccharides are produced by the bacteria, make up about 50 to 95% of the biofilm. So a major role in maintaining the integrity of the biofilm is protecting the bacterial from external attack. So this is why antibiotics and everything can't really penetrate into these things to get to the bacteria underneath, because that slime matrix is really acting is really a shield to protect these. It acts as a buffer, assisting retention of extracellular enzymes, enhancing utilization by the bacterial cells. So let's talk about biofilm biology. And this basically, and we'll go into this a little more depth, this is basically, if we go from left to right, this is basically the process we have a tendency to see. We get attachment and colonization of bacteria, they move into a site. Let's say that first molar, they work their way into the buccal sulcus. We get growth and proliferation, the biofilm grows, it matures, pieces of it detach, and they move elsewhere in the mouth or elsewhere in the body to have their effects. So onset and progression of disease, bacterial colonization is determined by the availability of the bacterial species and the co-aggregation pairings between them. Specific co-aggregation partners have attached previously. Biofilm formation is not random, but follows a specific pattern of succession. Early colonizers predominantly streptococci attached to a conditioning film composed of proteins derived from the saliva or clavicular fluid. Subsequently, actinomycetes and fusobacteria bind by co-aggregation to these pioneer species. It's basically another example of a biofilm in a sulcus. We can see various different bacteria in there. So adaptation through genetic improvements, horizontal gene transfer occurs, bacteriophage transduction, the bacteriophage carrying the DNA of the donor bacterium absorbs to a recipient bacterium. Conjugation, genetic recombination, in which there is a transfer of DNA from a living donor bacterium by cell-to-cell contact. In gram-negative bacteria, this typically involves a conjunction of cell pilus.
All right, folks, time for our second poll for this evening. We're going to let Dr. Kurtzman grab a quick glass of water. We're going to open our second poll. We'd like to understand what aspect of biofilms, as you think about your university experience and your future career in dentistry as a scientist, et cetera, what is the most interesting to you? Is it the pathogenic bacteria? Is it quorum sensing? Or is it the interactions and functions and the compositions of the biofilms that Dr. Kurtzman's been touching on for the last segment of the presentation? Always interesting. And in fact, folks, your opinion matters a great deal to us in this scenario, because we will incorporate some of this feedback in the future segments of the webinar, as we're going to be touching on some related topics that Dr. Kurtzman is going to introduce just at the tail end of our presentation tonight, but including things like cardiac health and women's health and oral cancer and diabetes for certain. And understanding how your perspective we can help address that, will continue to shape the presentations for you expressly to respond to some of the things you're interested in. All right, leave it open for just another moment or so. If you haven't had a chance to vote yet. Okay, everyone. And here are our results. All right, Dr. Kurtzman, we're going to turn it back over to you.
Thank you. So maturation of the biofilm. Formation includes a series of steps that begins with the initial colonization of the pellicle and ends with a complex formation of a mature biofilm. Biofilms exist in a variety of tooth surfaces, including fissures, smooth surfaces, and within the gingival sulcus. Biofilms are most likely to be seen in their mature state in periodontal pockets, as these areas provide protection from removal by home care by the patient. And one thing I'll touch on here is, and I'm not sure if they're giving this in school, but the research shows that anything deeper, a pocket deeper than 3 millimeters, a patient can't really get in there with floss or a toothbrush to really clean those areas. So anything that's deeper than that, if you have a 6 or 7 millimeter pocket, you're going to have something down there that they can't get on a daily basis in there to clean. So as the biofilm matures, its microbial composition changes from gram-positive to a structure filled with gram-negative anaerobes. And basically, anaerobes don't like oxygen. They're in an oxygen-poor environment, and they thrive. And most of the issues we have are really related more to anaerobes than aerobes. The first step in biofilm development is absorption of host and bacterial molecules to the tooth surface. The resulting pellicle acts like an adhesive sticking bacteria to the tooth surface. And we all know that basically plaque is very sticky. That's what basically holds everything in place. This early biofilm is able to withstand chewing, swallowing, ciliary fluid flow. And those early colonizers are also able to survive in the high oxygen concentrations present in the oral cavity. This initial biofilm always presents orally, forms immediately after cleaning. So basically, if you clean, within a short period of time, it's already starting to build up again. And it's hard enough getting patients to brush twice a day, much less continually. Co-adhesion of later bacterial colonizers in the initial biofilm continues to involve specific interactions between bacterial receptors. Those interactions build up the biofilm and create a more complex and diverse environment. And these diverse bacterial species create synergetic and antagonistic biochemical interactions among the inhabitants of the colonies. This may contribute metabolically with other bacteria that are located physically close to them. The bacteria continues to divide until a 3D mixed culture biofilm forms that is specifically and functionally organized. Polymer production causes development of extracellular matrix, what we're referring to as a slime matrix. This matrix is a key structural aspect of the biofilm, offering the inhabitants protection from the external factors. As the biofilm thickens and becomes more mature, these anaerobic bacteria are able to live deeper within the biofilm. And that's basically a good synopsis, an image showing this. We get initial attachments, we get irreversible attachments, it progresses to that, and we start getting maturation where it starts growing, and finally it gets to a point where it basically breaks off, bacteria break off with slime matrix, and they basically transport to other areas. As we said, it could be to adjacent sites in other teeth, or it could be elsewhere in the body. So bacteria invade host cells. Human cells have small openings where materials are passed to the environment, and that's where the bacteria enter. And that's a good example. Bacteria is shown entering a portal in a cell, carrying along those bacteria attached to their cell wall. Note how bacteria invade the host cells and avoid the immune system response. The body can't react to it if it's sitting inside a cell. Bacteria are chemically attracted to the cell openings and move from the periodontal pocket to the inside and carry attached bacteria with them. So they're basically able to invade through the gingival tissue and get out into the system that way. Oral bacteria through exopolysaccharides and exotoxin initiate a host signal response. Some aggressive bacteria are capable of penetrating human body cells. These bacteria secrete a toxin that causes major alterations to the surface of host cells. And these pathogens are then able to overcome the cell's defense mechanism, making it easier for them to penetrate the cell. Genes of some of these, like gingivalis W83, differentially expressed during invasion of primary human coronary artery endothelial cells. Analysis of microarray images indicated that 62 genes were differentially regulated. Of those, 11 were re-regulated and 51 were downregulated. The differential expression of 16 selected genes were confirmed by real-time PCR. Research supports that the effect of oral pathogens, especially as it relates to inflammation, is having an effect on the host distant from the mouth. Harmful strains of bacteria in the oral biofilm can enter the bloodstream through the inflammatory response and travel to other areas of the body. When we have a periodontal issue, what's the first sign of it? Bleeding. This basically gives those bacteria an avenue into the blood system that allows it to transport throughout the system. This exerts distant systemic effect that has been linked to numerous diseases, including coronary artery disease, cardiac disease, respiratory diabetes, many others that we'll cover in parts 2 and 3 of the series. Evidence indicates that patients with periodontal disease have a much higher risk of developing CVD and other systemic issues than those who prevent measures to control the biofilm in their mouths. I'm a strong believer in genetics. I think that there are some things we have no control over and some things we do, and the things that we don't have control, we can mediate the response to those that our body is having. And part of that is a healthy mouth leads to a healthier system. So in summary, biofilm is a complex material functioning through coordination of bacteria with a protective slime matrix. Extensive research has demonstrated that oral biofilm causing periodontal disease has distant systemic effects, has been connected to numerous medical conditions. The health conditions introduced here are not the only ones associated with periodontal disease, and review of additional conditions, diabetes, women's health, and so on, will follow in subsequent webinars. And periodontal treatment is evolving to be a more major component of full-body medical care, and controlling the associated biofilm yields better overall systemic health. We take better care of our mouths, especially as we're getting older, we can lessen and maybe eliminate some systemic effects that are happening. Proper home care is critical to biofilm management due to the rapid rebuilding of the biofilm following professional debridement. Brushing and flossing and oral ligation is often ineffective due to patient compliance and the patient's inability to effectively reach the bottom of the pockets. As we said, you can't really get beyond 3 millimeters with most home care, and oral irrigators are not much better. Professionals must carefully consider what oral health instructions they recommend to patients for best effectiveness and compliance. Any questions? We'll leave that up to Rich now.
Right. Thanks, everyone, for bringing those questions to the front. We've got a number that are coming in already, and appreciate the chance to tee them all up to Dr. Kurtzman. Let's just start with this, Greg, if you wouldn't mind. You've spent obviously a lot of time studying the literature. This summer you had a really great piece that came out in one of the industry's publications. Let's start with this question just simply. Whose work and whose research is influencing you the most these days? Who are you reading and building your information sources from that's really striking a chord for you?
Well, you know, it's interesting because it seems like not a week goes by where I'm not opening Facebook or someplace else where there's something that somebody's posted from the research linking some systemic effect really basically to oral biofilm. And we're seeing, as I said, we're seeing this on an accelerated basis. We're seeing more and more stuff coming out. And it's interesting, a lot of the stuff is in the medical journals. So I think as dentists, we have to start educating or communicating with our medical brethren to say, hey, look, you know, we're treating the whole system. We're just treating part of it, but it's connected to yours. Your patients maybe that have, maybe they're having trouble controlling diabetes, maybe they need to see a dentist to get their periodontal health in better control that oral biofilm, so we basically can get a handle on everything. But I think that, you know, there's nobody in particular. I would say there's just so much literature coming out these days.
I'm going to spring ahead to a question that Michael had posed, saying that you had remarked about research and the role of the medical community. How do you share that information as a practitioner these days? Is it a study club environment? I mean, to what extent do you see some strategies for doing that yourself in practice?
Well, what I've done is, you know, I have written a few articles. So what I do is I send that out to some of the medical colleagues out in the area just to educate them. And, you know, you can do that yourself. Basically, just see an interesting article. You send it out to a physician or people that are in your area, maybe the physicians that are treating some of your patients, to educate them. Because it's a marketing process. Because you're basically saying, here I am, I can help you treat your patients. You're not doing it, sort of works together. Here's some information. The problem is that you really have to boil this down into almost bullet points, because most of the physicians are bombarded with so much stuff and dealing with insurance companies and everything else. They don't have a lot of time to read stuff. But if you give them the basic information, I think that it basically helps educate them that we're more than tooth mechanics. We're there as healthcare providers.
For certain, for certain. Let's touch back on our poll question that started out this evening, and the interest of our attendees tonight on the conversation that takes place with your patients. Let's start with this one that was posed by Jill during that time frame. Who conducts the conversation? Is that a conversation that you have with your patients? Is it the dental hygienist? How do you divide that conversation up in your practice?
Well, I usually have the hygienist basically starts the conversation because they're really seeing them first. And then when I come in and do the exam, they're basically saying, oh, I see these issues in this patient, some bleeding, some pocketing. So then I pick up on that. And, then I'll say, hey, I realize that you have some pulmonary issues or diabetes and everything. Some of this may be linked to better taking care of your mouth. And especially, I'd say probably about 30% of my practice are geriatrics. And a lot of them have a lot of health issues. And when you start saying to them, it's like, if you take better care of your mouth, do a better job brushing, it may have a positive effect on the rest. You start building stuff in them. And when you basically give them information in a sense, and you're not trying to sell them something, I'm not selling you this product, I'm just giving you information that may help you take better care of yourself, I think they tend to be more receptive to those. And then you get some patients who say, look, hey, I had perio surgery a couple of years ago, and it didn't do anything. And of course, you say, well, how often have you been to the dentist since then? I go, oh, I haven't been to the dentist. I go, well, that's the thing. It's like buying a car and never changing the oil. You got to take care of these things. So a lot of this, what we do in dentistry, is really educating patients as to what's going on. I believe I give them some information, and depending on the reaction, if they ask questions, I give them more information. If I'm really butting up against a wall, I basically just sort of end it like that, and I say, all right, let's see you in 6 months, or some patients that say it'd be better 3 months, and you start building on that. And sometimes you have to see a patient a few times and repeat stuff before the light bulb goes off, and they go, you know what, I am having an issue, maybe it's time for us to do something about it.
Fully charted, I'm sure, noted in the patient's chart, all of these type of conversations you're having with them, the regimen you might be prescribing them, all part of that dialogue, part of the systematic approach you apply every day?
Yes. The key basically, as Rich touched on, is really it's documentation. If you're basically treating a patient and they say, no, I don't want to do scaling or root planing, I'll just come in every 6 months, and they ignore it, you write it down, because if what happens is their insurance changes, and they go to another dentist that's now on a different insurance plan that you're not with, and they go, wait a minute, you've been seeing Dr. So-and-so for 5 years, you got this really bad perio problem. How come you didn't? And of course, patients never remember. You have documentation saying, hey, I saw this person every 6 months, I recommend a scaling or root planing, I recommended this, that, and the other thing, I told them they should come in more frequently. They basically declined every time. So you're putting the responsibility on the patient. You can only offer them information and a service. It's up to them to follow through and do something. But the key is documentation.
I have a new question here. Do you change your approach diagnostically for a patient that's maybe more at risk, or do you conduct the same systematic approach each time?
I basically do the same approach each time, but if I see that somebody has issues, and I'll usually, when I come in, it's a new patient, I'll look over the health history and I'll ask them, hey, how well was your diabetes controlled? Do you have this problem, that problem, based on what they've indicated? And then basically I start going from there. So we sort of start with a basic thing, and then we customize it depending on what the patient's conditions are, and as I said, how receptive that patient is to the information. Some people don't want to do anything, and some people say, hey, I do want to do something. And I always tell patients it's cheaper to maintain than it is to treat later on. So we can do something, get you in better shape, and it may have a positive effect on your general health. But if we ignore it and it gets to a certain point, dentally it might be very expensive at that point. We're losing teeth. We're doing a lot of laser treatments and stuff to really solve the perio problem. Or systemically, now you've had a lot of issues with that. You've developed, pulmonary issues. Your diabetes got worse. That's taken an effect on your system. So basically, you're sort of telling the patient, we're here to help you, but it really depends on what you want to do. And as I said, some patients say, yes, I'm ready. I have a patient, perfect example, was in 2 weeks ago. I've been telling him for the 3 years I've been treating him, he comes in every 6 months, you need scaling or root planing, this tooth that's loose needs to come out. And he came in 2 weeks ago and he said, oh, that tooth that you said is loose? I go, yeah, I figured he's going to say, I'm ready to have it extracted. He goes, it fell out the other day. I'm like, okay, his teeth, I don't think this guy uses a toothbrush between visits. And his attitude is, I just don't want to do anything. And, so there's nothing we can do. Some people have the attitude, you tell those patients, I don't want to have you in my practice. I don't want to do that. I'm basically going to try to educate them, and hopefully they're going to get to a certain point where they're going to say, all right, now I'm ready.
I know that we're going to get into in-depth in one of our future sessions, Dr. Kurtzman. I know we're going to talk a little more about management. We have some questions here that are coming in that are related to, let's say you have that patient that has that 3 millimeter depth in their pockets. What's your standard approach for that patient, maybe compared to what's your professional intervention, your approach for someone that does have 7 millimeter pockets and a greater progression of the disease?
See, unfortunately in dentistry, we've sort of educated patients that bleeding in your mouth is okay. So they might not have pockets, but they're getting bleeding when they brush, and they go, that's normal. I always say to the patient, if you brushed your hair, of course this doesn't apply to you, Rich, but if you brushed your hair and there was blood on the hairbrush, would that be normal? And they go, of course not. I go, then why is bleeding in your mouth normal? Bleeding anywhere is not a good sign of any health. It's a sign that there's a poor health issue going on. And I basically, say, all right, this is what we got to do. And it may be, increase, you see them more frequently. I like to recommend electric toothbrushes. There was a study done many years ago, and I think it was Sweden, and they said, what's the ideal amount of time somebody needs to do to brush their teeth? And I think the answer was like 5 minutes. And then there was other studies saying, what's the average time that people actually brush their teeth? And it's less than a minute. So if you can make it more efficient in that time, because it's easier to increase efficiency in a time versus telling somebody they got to brush longer, then I find that they're going to do a better job. And electric toothbrushes are better than manual, and they've gotten very inexpensive. So I'll try to steer people to that. I try to get people to floss, but the problem is, and I remember there was a guy who worked for Butler many years ago, and I asked him one day, I said, how come you never see ads for dental floss? And he says, well, only 5% of the dental population has teeth actually flosses on a regular basis. We're not going to market 5% of the population, where half of the people use a toothbrush. So that's why you see toothbrush commercials. You don't see anything for floss. So basically, I'm going to try to encourage people to floss. Most people just don't have the dexterity. There are floss aids they can use. I try to improve the home care they're going to do and give them some hints on how to do this. Then we start moving into basically, hey, you got some pocketing. We're going to have to do some deeper cleaning. I really am not a big believer. We used to do a lot of flap surgery. That was the standard. You had pockets, you flapped it, you cleaned it out. We found that, especially with lasers, we don't need to do that anymore. We can basically go in and scale and root clean under local anesthetic. We can use a laser in those deep areas, and we can get some regeneration. The key basically is getting that patient into a stable position and getting them to the point where they're learning how to do home care. Because face it, they got into that position because they weren't doing home care. They weren't, whether it's laziness or whatever, we have to retrain them. And the key sometimes is, do you want to keep your teeth? I tell them, I have patients that are almost 100 years old. I had a patient in 2 weeks ago who's 99 years old, and she's probably been a patient for 25 years, and she's missing one tooth, and that's it. And I said hey, everything's looking good. And she said to me, he goes, well, that's because you've been doing such a great job taking care of it. So people can maintain their teeth for a long time, and I think a lot of this falls on her. She does a great job at home. But the key is, everybody, if you take care of your teeth, most people can maintain their teeth for life. And if you're missing some teeth, if you still have teeth, you can maintain those. The key is just changing your habits.
What about hydrogen peroxide, Greg? I know that there's a certain degree of aspect when you're trying to deliver that into the pockets on a professional level. That's got some complications, though, doesn't it?
Well, the problem is with peroxide. Peroxides are great for breaking down the slime matrix of the biofilm. The problem is that the sulcus is producing fluid, and Mother Nature puts it that way, so it pushes any foreign bodies and stuff out of the sulcus. So the problem is, if we try to irrigate in, if we rinse with it, it doesn't get into the pockets. If we try to use an irrigator to push it in, it basically gets pushed back out by the sulcular fluid. And it really is contact time. Stuff has to really be in contact for 10 minutes with the biofilm to break it down. So there are basically some, and we'll talk about that in part 4, there are customized trays where you can use peroxide gels, where it's sort of holding it into the pocket. It's resisting that clavicular fluid flow. And it basically is allowing that contact time sufficiently so that it can break down the biofilm, and we can basically get rid of the biofilm.
Interesting. All right, folks, we have a few more moments if you have any last questions. We've been able to have a pretty good stream here with Dr. Kurtzman since we finished the didactic portion.
I'm an ex-New Yorker. I talk fast.
And have the same alma mater, coincidentally.
Yes.
Once upon a time.
But we're off by a few years.
Yeah, I was there first, right?
Yeah, I think so. So
all right, folks. Yeah,
any other questions anybody has?
It looks like we're just getting to the end of the Q&A. Okay, so let's talk about what's next on the horizon.
We're going to basically, on October 17th, we'll do part 2. We're going to talk about systemic effects of oral biofilms, cardiac, erythemosis, kidneys, pulmonary, diabetes, colon, and oral cancer. And part 3 we'll do on November 7th, and we'll do some more systemic effects, concentrating on prostate, pancreas, liver, pregnancy, fertility, Alzheimer's, and artificial joints. And part 4, we'll bring it all together. We'll talk about how do we recognize this and how do we treat it. It's great having this knowledge, but if we can't apply it to actual treatments, and we'll talk about easy ways because it's a compliance thing. If we make it too difficult, patients don't do it. If we make it easy, then patients will actually do it, and then it makes it, it helps them and gets the, we achieve the goals that we want.
I have a quick question for you before we break.
Sure.
Of all these different areas and facets that peristomach is today in the practice, what's the most interesting to you in your future exploration of the topic? Where's your focus now?
You know, it's interesting. I volunteer at one of the national parks, and one of the other volunteers were at a picnic for the volunteers on Saturday, and she was complaining. It's like, you know, her diabetes, she's having a lot of trouble controlling it. And she said her dentist told her that she's got some deep pockets. And I said, you know, here's something that I'll throw out to you. Maybe it's your perio issue is maybe messing up your diabetes. That's why you can't control it. Maybe if we get your perio under control, maybe that'll help with your diabetes. So I think that basically I'm trying to educate people. I'm trying to get them to get on board with taking better care of themselves. And hopefully, you know, we're having an effect on people's lives. As I said, I'd see about 30% of my practice are senior citizens. Some of them are nursing homes. And the problem with that is they're getting very poor care. So it's trying to educate the caregivers that they have to do a little better job helping them keep these things clean so that they don't have their patients becoming more systemically ill and affecting the quality of their lives.
Dr. Kurtzman, I just had one more that came in under the wire as well.
Sure.
What's your recommendation for oral irrigator use? And do you recommend that in combination with floss?
I mean, floss is really good for getting rid of food that's stuck between teeth. Oral irrigators, like I said, and we'll talk about that when we actually get to part 4, they're semi-effective, but they can't get into real deep pockets. They just, if you turn up the force high enough to get it down there, it causes damage. So if you keep it at a point where it's not causing damage and forcing food particles in, it really is not effective. They really have limited use. I think that, you know, years ago, water picks were the big thing and other similar things, and they've sort of fallen out of disfavor. You don't hear those mentioned as much because they're just not as effective. And when you use them, you know, your spouse yells at you because there's now stuff sprayed all over the bathroom that wasn't supposed to be there. But, so I think that
that might not be in the science, Dr. Kurtzman.
Yeah, I don't know. I try not to get my partner upset at me with those things. So.
Fair enough. Well, thank you so much for spending your evening with us today. We really appreciate it, all the insight and the time that it took you to assemble not only the presentation, but the knowledge that's necessary to deliver it as effectively as you did tonight. So on behalf of our audience here in the next EDS, a big round of applause for you, sir. We'd like to thank all of our attendees for taking time out of your evenings and your studies to come join us, and certainly to our sponsors at Sunstar for helping to bring an informative program like this forward. We really appreciate their support and couldn't do that without them. We're going to close the presentation. If you wouldn't mind, I'd love for you to stick around and just complete this brief survey. We're going to use it to, again, start to fashion the next aspects, parts 2, 3, and 4 that start to kick off in just a couple of weeks, as Dr. Kurtzman mentioned. So thank you all for your time, and a special thank you to Dr. Kurtzman, and we look forward to seeing you on October 17th for the next webinar in the series.
Yeah, if you have any questions, feel free to email me. Thank you.
All right. Thank you so much, sir.